So what I want to talk about today is the electrical power supply. It's very simple. So I want to say two things. Life is an emergent property, and when I talk about an emergent property, let's be sure we understand this in a simple sense. So for the last 50, 70 years, we've made parts lists. So imagine you go to the grocery store and you fill up the grocery bag with all kinds of stuff. You have some fish, you have some vegetables, you have all kinds of cans of tuna, and then you go home and you jump up and down on the bag and you break everything in the bag. And then you put it in the centrifuge and you say, what did I buy? That's basically the way we've treated cells or organisms for the 100 years almost. And now we have omics. So we have genomics, we have proteomics, we have whatever omics you want. So we've made parts lists. So here are the parts. Here are the parts. I'm telling you, we know the genes of an organism. Very simple. Okay, make me the bug. No can do. So far, it has not been done. And that is the problem of an emergent property, that we do not know how to think outside of the hypothesis of decomposing something into its parts. Reconstructing things from its parts is something that's very different philosophically. I'm just going to leave it like that for a moment. Now, obviously, organisms derive energy for maintenance by moving electrons. You're doing it right now as you breathe. You're breathing in oxygen, you're breathing out carbon dioxide and water. You've just reduced the oxygen to water, basically, and you've oxidized the carbon. So all substrates and products must ultimately be cycled, which means on a planet you can't just have one bug. So biological processes are paired. And that doesn't mean that they can go backwards and forwards. So you're an oxidizing organism. The plants outside are reducing organisms with respect to carbon. They're not the same pathway going backwards. That's very critical to remember, because thermodynamically it's critically important. So here is something I wrote a paper about with Tom Fenchel and Ed DeLong many years ago. And it's just, we just put together what I call the electronic diagram of Earth. So we have a very simple diagram. I've left out a few things here. I've left out arsenate and selenium and crap like that. But the point is that we can draw the electronic diagram. So these electron flows are mediated by organisms. I don't care what the organism is, it's a metabolism. And the metabolic map, turns out I was wrong in the paper in 2008. I guesstimated about 1,500 genes. I tasked one of my former graduate students, Ben Yellen, to go and actually do the arithmetic, go into each one of those pathways and discover the genes. And there are only 400. So there are only 400 genes on the planet that really move all the electrons around. That shouldn't be too hard. We're not looking at 10 to the 5th. Now, when we think about metals and transistors like this, the way I think about it, because I was trained originally in electrical circuits, I think about it as an electrical circuit board of the entire planet. So we have one power supply, that's the sun. We have two wires, two main wires, the atmosphere and the ocean. Those are the wires that are moving the electrons around between organisms. And everything else on that, every bug on that circuit board that's moving the electrons around is not really a transistor. I'm going to explain to you in a minute. There are diodes. A diode moves an electron in one direction. It can't go back and forth, with very few exceptions. So here is another way of looking at this. These are the intersections of metabolisms. Metabolisms are interdependent. You can't have just one metabolism on a planet. They're interdependent. And what you see in the center there of one of these is the TCA cycle, the tricarboxylic acid cycle. That's one of the most ancient and most important metabolic pathways existent. It was actually originally the reverse TCA cycle. We can talk about that later on if you like. Now, let's talk about this. This is the very funny part. So we do not have a phylogeny of metabolism, per se. So we have ancient proteins that we can postulate, which contain maybe 100 atoms or 200 atoms, very, very small. And then you have these gigantic molecules, which are the extant manifestations of these pathways. For example, nitrogenase. Nitrogenase is absurd. How did we get nitrogenase? It's a singleton. We don't know. And as we'll see that some of these other really, really important pathways, we do not understand their origin, like oxygenic photosynthesis. It's a singleton. So let's talk about the core motifs or structures. And I want to just remember, or have you remember, the Eck and Dayhoff. Margaret Dayhoff was the mother of bioinformatics, and they wrote this seminal paper in 1966, which was published in Science. And it says, the processes of natural selection severely inhibit a change in a well-adapted system on which several other essential components depend. What does it mean? It means that when you have a metabolic pathway, that the core of the pathway, the core machinery, hardly undergoes evolution. It is very stiff. So let's just talk about this for a second. We have oxygenic photosynthesis and aerobic respiration, as I said. So there's movement of electrons in opposite directions. The one on the left, the oxygenic photosynthesis, requires a driving force, and that is light. The one on the right uses that driving force. Every single covalent bond in your body is coming from the sun. It's amazing. You're just a stored, you're a battery that has been using the solar radiation from oxygenic photosynthesis. Now, in information theory, and I'm not going to go into the Shannon information theory story here. We can talk about that another time, and Eric probably knows much more about this than I. But the idea is that if you have an amino acid sequence, you can have several types of folds or secondary structures or tertiary structures ultimately. So you can have a spaghetti, then you can make a rotini, and then you can make a lasagna. Those are the three main folds, okay? So you just have to know pasta, and now you understand secondary structures of proteins. Now, let's just take a look. This is Zhenren Shen, who's at Okayama University. If you don't know him, you should invite him here if you've never had him. Have you invited him? He's an amazing person. Zhenren and I started in 1985 in Japan when he first came from China. So what we have here is the photosynthetic apparatus that splits water. And the core of that apparatus is a cubane. So where's my pointer here? It's here. Yes? Okay, so it's a 4 manganese, 1 calcium cubane. All right? This is a singleton. That metal motif is not found in any other protein on the planet, and it's not, to our knowledge, found in any mineral. Without that cubane, and we've done many experiments where you can substitute different metals here, you do not split water. So this is the essential component of the water splitting process. And now you say, okay, so Falkowski, when did we start splitting water? And the uncertainty is from about 2.5 billion, just before the Great Oxidation Event, to about 3.4 billion, or even a little earlier. So it's about 1 billion year uncertainty in the evolution of oxygenic photosynthesis. I'm not talking about when we oxidized the planet. I'm talking about the process that led to the oxidation of the planet, incorporating as well plate tectonics, because if you don't bury the organic matter, you don't oxidize the planet. I'm not going to go into that. That was another lecture for another time. But you can see how complicated these proteins became. So you have many transmembrane spans. It's a dimer, a complete dimer, and inside is a dimer of dimers. So the closest relative to this is a purple non-sulfur bacterium, which cannot split water. It's an anaerobic photosynthetic organism. So, take-home message to start. Metabolic pathways are not simply reversible. We don't have this structure for you to oxidize carbon. Okay? That's not in your body. So they form electron transport chains based on thermodynamic disequilibrium, and essentially all organisms are diodes. Now, when we talk about thermodynamic disequilibrium, what do I mean? It means that we're differentiating the Gibbs equation with respect to time. Now, two things. The second thing that's really essential for metabolism is metals. So here is one protein, and you can see that there are little bits of metals inside, and we're moving electrons through here by tunneling. And this follows what's called the Moser-Dutton rule. These metal proteins, these metal motifs, cannot be more than 14 angstroms away from each other, otherwise the electron is not going to move. It'll stop. So 14 angstrom is the magic number of the maximum distance between these molecules within the protein. The protein is a scaffold. Okay? So we're moving electrons within a protein. There's an electron transport chain within proteins, not just between. And now let's just take a look very quickly on Earth. And this is an old figure from one of my former postdocs. This is the amount of iron in all of the electron transport moving proteins called EC1 proteins, enzyme commission 1, the oxidoreductases. So almost 2/3 contain iron, either as heme much later or as iron itself, as iron sulfur, for example. And then we have copper, which came in later after the oxidation, more or less after the oxidation event. And then you have a little bit of manganese, a little bit of nickel, a little bit of molybdenum. And I'm just going to say this. There's an incredibly interesting paper that if you haven't read it, or if you're a graduate student, if you haven't read it, you certainly should read it. It's by R.J.P. Williams, 1982, I guess. It's called The Bakerian Lecture. The Natural Selection of the Chemical Elements. Not the book, the paper. It was one of the most profound papers that has influenced me for 25 years. Bob Williams was an amazing, amazing chemist at Oxford. All right, now, let's just take a look at how we get from these different states in metabolism. So here we have hydrogen is probably one of the earliest metabolic pathways, and we have this as a gas, which is not in the atmosphere anymore, but it was relatively much more abundant, at least in the oceans, on early Earth. And then we get to the so-called great oxidation event, and this is in log scales. So you have to look up, this is PAL. And so we started to oxidize the planet around 2.35 billion years ago, somewhere around there. And we got up to about somewhere in the neighborhood of maybe 1 to 3% oxygen in the atmosphere, maybe even less. And I'm not going to go into how we know that, but it's obviously a little more arcane. It's from the mass-independent isotopic fractionation of sulfur and then the oxidation states of various metals, transition metals. And then we get into an oxidized world. And then we get a second burst of oxygen with the rise of eukaryotic algae and the rise of trees. And we start to have only oxidized species, as Renaissance said. Now, the first metabolic reactions were catalyzed by small metalloproteins. That's an hypothesis. If you don't have a transition metal on the protein, we can't move electrons. So there are no fossil records. There's no phylogeny of this. So we have to really think about how to do this. And so at Rutgers we have, and we've had actually at Brookhaven before I came to Rutgers, sorry, that's Steve Burley in the background. Steve Burley is the director of the Protein Data Bank. So we have about 210,000 structures to date, and we have all of these protein structures in the Protein Data Bank based on crystallography. And we also have a set of proteins, and this was something I pushed a few years ago, which was met with quite a bit of resistance from some of my colleagues, to be honest with you, because we can predict protein folds now pretty accurately. So when you go into the protein data bank, if it hasn't been elucidated, but you have the sequence, you can estimate what that protein structure is pretty accurately. Now, based on whether it has a lot of spaghetti or rotini or sheets, so this is the lasagna clade, here's the spaghetti clade, and here is the rotini clade. Okay, you can see we can go way, way, way back, and you find iron-sulfur clusters in the earliest hydrogenases, and they are also in ferredoxins. So that fold, so-called fold, is called the ferredoxin fold. It's CXXC, XXC, XXXXC, like that. The CXXC, XXC is critical. So the cysteines are binding to the irons. So we can dissect these megaproteins today, the extant proteins, and we can look at these different groups, and we can then find out what the Legos are that make them up. And let's just go down through this. So we have modules of these, and we can dissect them out, and we can have to make them less than 14 angstroms. And what's called a spatial adjacency network span, which was developed in collaboration mostly by Vikas Nanda, one of my close collaborators who was a student in Dutton's lab. And then we can start to do some very, very simple things. So here is the real extant ferredoxin, which Eck and Dayhoff worked on. And then we can make a very, very, very small iron-sulfur cluster with DL, 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 cysteine, 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 and let's say you can put arginine, doesn't matter. Anything in there to make it spatially arrangeable like this. And you can make an incredibly small ferredoxin, which is incredibly stable. You can go 1,000 times around in cyclic voltammetry, and that thing will just transport electrons and transport electrons. Very, very, very simple. So let's keep going on. Let's go on to a modern oxidoreductase, and this is the nickel-iron hydrogenase, which contains iron-sulfur clusters. Okay, now it also contains nickel. So we can now make or design a 13 amino acid dual nickel hydrogenase. 13 amino acids. It's incredibly stable from pH 5.5 to 8. Thousands and thousands of times it can go round and round. You don't need that big monster protein to make a hydrogenase. You can make hydrogenases from very, very, very simple molecules. And it's incredibly robust. The turnover time approximates that of the natural hydrogenase. All right, so now let's talk about origins. So here's where we spent most of the last 50, 60 years in prebiotic chemistry, making the amino acids, making whatever it is that you needed. And now we'd like to make very, very simple proteins that connect to each other to make a metabolic transfer chain. So I can put, for example, a source of electrons for my nickel back here as a ferredoxin that has only got 13 or 12 amino acids. And I'd like to connect that to an artificial, incredibly simple nitrogenase. Now we start to make a metabolic pathway. It's not a cell, but it is a power supply or a source and sink of electrons. And now it turns out that if we go into all those proteins, that we can find two main folds. There's one, that's the Rossmann fold, and there's a ferredoxin fold. We have a plastocyanin type fold and some erythrin fold here, but don't worry about that. So these two folds, the ferredoxin fold and the Rossmann-like fold, are the two main folds at the beginning of life. So, okay, so there are 12 core structural motifs, and in the first 2.5 billion years of history, nature invested heavily in R&D from which a core set, not necessarily the best set, of metabolic machines evolved. This is the Microsoft world. You write 5,000 pieces of code after you make the code to make this corrections for the code. So all of these were developed in prokaryotes. If you're studying eukaryotes, it's a waste of your time, in my opinion. And there are approximately 400 genes, and these metabolic sequences are coupled on planetary scales. And all of the contemporary electronic potential is driven by light. It's not coming from hydrothermal vents. The hydrothermal vent story is an interesting one, but if there was no electron gradient between the outside world and the hydrothermal vent, you would not have a redox gradient. And I like Mike Russell, and he's a good friend of mine, but we have a very, very long set of arguments about that. So these are the things that spark the life, short peptides in a metal. Chemistries are possible with these peptides. The early enzymes were actually very robust, and perhaps simple peptides are universal. So as per Vernadsky, for those of you that remember Vernadsky, all living organisms exchange a gas with their environment via redox reactions. And so the core processes are related to gas exchange. So if we're going to look at extrasolar planets, and there are changes in the gas composition that are far from thermodynamic equilibrium, for example, nitrous oxide and methane. It's very, very difficult to imagine nitrous oxide and methane existing on a same planet. They're so far in disequilibrium. This is one of Sagan's early arguments, Carl Sagan. And by the way, just as an aside, Vic Nanda's son is named Sagan. So we're looking at glows from other planets very poorly, actually. We're looking at what's called limb experiments for the most part, even with Webb telescope and some of the other telescopes. So we're looking at the little bit of gas as the planet is going in front of its star to try to infer the gas composition. So I'm going to leave you with this. Here are the websites. We have many, couple of hundred publications. We've been funded very generously by NASA Astrobiology and by the Rutgers Energy Institute. And there are my colleagues in crime. And many of them or some of them are here or have been here. So Nanda, Nathan Yee has been here. Shauna is here today, and so on. And I just want to say thank you.